Okay, welcome to the lecture entitled DNA Discoveries. A quote from Stephen Hawking. Science is beautiful when it makes simple explanations of phenomena or connections between different observations. Examples include the double helix in biology and the fundamental equations of physics. It's that simple elegance of it that really is where the beauty's at. So we already know in the past about macromolecules and these biomolecules. We talked about some of the chemistry of it. Now we're talking about some of the purpose. So in order to be living, some of the things that living things do or metabolize, it's using energy, grow. They have to respond to stimuli like different things that happen in the environment, reproduce, be able to adapt to their environments, they have to respond. So what DNA does is the instruction manual for all of these processes. It controls all of the building of everything. So in terms of metaphor, what's a metaphor you can create for DNA's purpose? Explain your response using the clear technique. Remember when we do clear, we're going to write a strong claim. We're going to provide evidence for that claim. And the reasoning is really the hard part. That's where we have to get into several sentences where we have to back up why that evidence supports that claim. So some terminology. Nucleic acids are the name of this group of molecules. That includes DNA and RNA. So that's what the NA of DNA stands for. So it's a polymer. So poly as in many parts. So these parts are nucleotides. The nucleotides are a relatively simple molecule. It's that A, T, C, and G that you've heard us talk about. So they're a kind of sugar. They have a sugar on them. They have a phosphate group, and then they have this nitrogen base. And the sugar and the phosphate is the same for every nucleotide of DNA, or every nucleotide of RNA has the same sugar and phosphate. What makes the nucleotides different is that nitrogen base. So we slowly discovered all the beautiful parts about DNA in a series of scientific experiments. And the scientists learned from the science before them and applied that information to use it to learn more. So let's talk about 100 years ago. Frederick Griffith. He had two kinds of bacteria, R and S, and was experimenting on mice. The R bacteria didn't kill the mice at all. The mice were okay when they had the R bacteria. Now the S bacteria was not okay. The S bacteria killed them. So in our graphic here, the R bacteria are the little circles, and the S bacteria are little stars. Stars are bad, circles were fine. Well, what they did is, with the S bacteria, they tried heating it up to destroy the bacteria. And that made it so that the mice lived. The mice could handle the destroyed bacteria. But here's where things got interesting. The destroyed S bacteria was okay. The living R bacteria was okay. But if you put together the destroyed S bacteria and the living R bacteria, it killed the mice. Even though separately the mice could handle both of those things. And so Frederick Griffith said, So what he called this was the transforming principle. He said that whatever it was that the S bacteria had that was dangerous, whatever it made them that made them disease-causing, he said even after you destroyed them as bacteria, the S ones, that the R ones were able to pick it up somehow. The R ones learned it from the S bacteria. And so he called this the transforming principle. He said that there is some kind of material that can be moved from one thing to another, and it tells it what to do. It gives it instructions on how to be what it is. So how do you think a discovery like that paved the way for the discovery that DNA is the blueprint of life? Fast forward almost 20 years. Oswald Avery, following up experiments about Griffith's transforming principle, did a series of tests on that transformation of how our bacteria could become S bacteria if you just put dead S bacteria around them, destroyed S bacteria. So he did tests and found that no proteins were present. And then tests were done that found that the proportions of elements match the same proportions of DNA. If DNA had X% of nitrogen and X% of phosphorus, then the unknown transforming principle showed the same percentages, which was suspicious. And to cap that off, he kept doing all these different things, and the transformation kept happening. That living R bacteria could become dangerous in the presence of dead S bacteria until he put in DNA-destroying enzymes. DNA-destroying enzymes prevented the transformation. So what was Avery's conclusion? That transforming principle, the idea that something could be transferred and take the information from one organism to another, DNA must be the vehicle through which the information was transferred. Almost 10 years later, we're in the 50s now. Alfred Hershey, Martha Chase. They studied a kind of virus, a virus that infects bacteria, called a bacteriophage. We have an image there on the right of a bacteriophage infecting E. coli. And so 2 experiments were done. They were done with 2 different bacteriophages. They had found a way to put different kinds of elements into this bacteriophage. So one of the types of bacteriophages, one of the types of viruses, they made it so that they had radioactive proteins. Specifically, they put radioactive sulfur into the proteins of this bacteriophage. Then in a different experiment, they put it so that this time the protein was fine, but they made radioactive DNA. And so bacteriophage 1 only had radioactive molecules in their proteins. Bacteria 2 only had radioactive molecules in its DNA. So they introduced, in 2 separate cases, bacteriophage 1 was given some E. coli. Bacteriophage 2 was separately given some E. coli. We know that viruses only transmit genetic material, like DNA, into bacteria. So what we saw later, the way we know this, is that the bacteria, after it was infected, in experiment 1 had no signs of radioactivity, which means none of the proteins were transferred. In experiment 2, all of those bacteria were radioactive because the DNA from the bacteriophage was transferred. So we advanced this by tracking the DNA. That's the only reason we used radioactive molecules, because they're easier to find, is able to tell which one is which. And so we followed DNA from the virus to the bacteria. And so that's how we decided the protein was not the transforming principle. The DNA was. The DNA was what's being moved from virus to bacteria. The DNA is what's encoding for what happens. The DNA is moving around the information. Okay, in that same decade, we have Erwin Chargaff. So he looked at the DNA of different organisms. And what he noticed, that of the 4 bases, adenine, thymine, cytosine, guanine, A, T, C, G. First of all, we see the same 4 bases in all DNA. Why? Because of common ancestry. But what he also noticed was that the proportions, whatever the percent he found of adenine, found the same percent of thymine. And the same thing happened with cytosine and guanine. So what that told us was that in this double helix, in the DNA, adenine must connect with thymine. That's why we see them in the same percentages, because they bond to each other. And guanine must connect with cytosine, because they bond with each other. Even now today, we have more information about the molecular structure of these atoms. We can see that A and T make a very similar pattern of G and C. You can see how it's always a double ring that attaches to a single ring. Okay. We also had another scientist. Her name was Rosalind Franklin, and she's the one who gave us the evidence that DNA was a helix, that it was a spiral. So we got some structural information about the shape of it. She used some specialized X-ray techniques that were used to X-ray at the molecular level. Okay, still in the century, the 50s were a very important time. James Watson, Francis Crick. So here we get all the information from all these people before them is all culminating in Watson and Crick's very broad explanation that they made about DNA. They didn't necessarily come up with every individual part themselves, but what they did was take all these different parts and add in all the details that made it one complete explanation. So, James Watson and Francis Crick told us that there's a double helix, that you have two structural components on the outside, like the edges of a ladder, and that those are all the same, no variation. But in the middle, between connecting those two edges, you get these bases, and they show up in pairs. And you always see A with T, and you always see C with G. You never see them connected in any other way. And it was that order of when you see ATs and when you see CGs, the order and the sequence determined the identity of that DNA molecule. So these base pairing rules are what allow us to see the information, to see what's going on, to track the code. The edges of those nucleotides, that sugar and phosphate that we talked about before, those covalent bonds form the edges of this ladder. And it's kind of like if you take a ladder and imagine you're able to twist the two sides until you wound it up into this helix. That protects the code in the middle. These two purines and pyrimidines that are hydrogen bonded together. The AT and the CG. That is where the code is at. So what did the word bacteriophage mean? That was a specific kind of virus that infects a bacteria. So we used it to track. We know that the bacteriophage was changing bacteria, but what was it giving them? What was the transforming principle? We use the experiment here to follow DNA. So what's the significance of this drawing? This drawing tells us two things. One, the base pairing rules. A goes with T and C goes with G. Two, it tells us the structure of the molecule that they're in, this wound up double helix. So every organism's DNA includes the same bases: adenine, thymine, cytosine, guanine. What does this similarity indicate about the origins of life? This is an interesting question because there's all kinds of different bases that could have been. These were useful because of their stability. They had a strong structural component, but ultimately the base is not the information. It's just a sequence of the bases. The fact that these four are the ones that get seen over and over is slightly arbitrary. So what does it indicate to us about the origins of life? There was a common ancestry. That early life, the biochemical pathways found adenine, thymine, cytosine, and guanine to have an advantage. organisms that used those had an advantage in replicating and in advancing. And we've inherited that advantage from those who came before us. There's been no advantage to changing. It's been so successful that, we see this commonality between, all life, that common ancestry.